Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on cleavage. Now, the process of fertilization we have studied in the previous lecture. So, on the screen, as you can see, I am showing you one secondary oocyte. For example, this one is the secondary oocyte, and this secondary oocyte is surrounded by many sperm. So, these are the sperm, and the cells which are present around the secondary oocyte, these cells are known as cumulus oophorus, cumulus oophorus cells. Now, we need to understand this thing, this cells, cumulus oophorus. See, this is the secondary oocyte, okay? For example, here I am drawing, this is the secondary oocyte, and it is surrounded by granulosa cell. So when this secondary oocyte inside the Graafian follicle, at that time this granulosa cell are known as cumulus oophorus cell. But when this Graafian follicle ruptures and the secondary oocyte is released, the same granulosa cell which were earlier known as cumulus oophorus, these cells are now known as corona radiata. This thing we have to keep in mind. So we can say now the secondary oocyte, these cells are corona radiata, and the entire secondary oocyte is surrounded by many sperm, and we know in the process of fertilization only one sperm is participating. Only one sperm is able to penetrate the secondary oocyte, and in the process of fertilization the nucleus of one sperm enters. Okay, so the nucleus is entering in the secondary oocyte, and this nucleus swells in the secondary oocyte. At this time, this is the nucleus of secondary oocyte, and it completes meiosis 2. So due to which the second polar body is produced, and this nucleus, I am sorry for this interruption. For some reason, I have to pause this presentation lecture. Now, we were discussing, this is the secondary oocyte and this is the nucleus. So, this nucleus is completing meiosis 2. So, the moment the sperm is entering in the secondary oocyte is completing meiosis 2. So, when the sperm nucleus is present inside the secondary oocyte, so we can say now the secondary oocyte is become egg, or now instead of secondary oocyte this cell is known as oocyte. Okay, now after some time this second polar body is going to degenerate. This nucleus is swelling, so this is known as female pronucleus. This sperm nucleus, it is also swelling, so this is known as, this one is a male pronucleus, this one is a female pronucleus. Both the pronucleus, they are combining, and the single nucleus is produced. So in this there are 46 chromosomes, and this entire cell is now known as zygote. This zygote is produced. This entire process of fertilization is taking place very close to the fimbriae in the uterine tube. So in the uterine tube there is a zygote, and now the process of cleavage is taking place in the uterine tube. So now we are starting the discussion of cleavage. Now, before our discussion, I would like to show you the diagram. This is the cell division of the cleavage, the image under the microscope, and this is the entire schematic diagram of the cleavage process. So, one by one, I am discussing all the points with you. So, what is cleavage? So, cleavage basically is a rapid mitotic cell division. Now, we have to remember this thing, mitotic cell division is taking place, and large number of cells are produced. But this point we have to understand, without intervening the growth. Now, what is the meaning of this? See, we are having zygote. From zygote, due to cleavage, 2 cells are going to produce, then 4, then 8, then 16 cells are going to produce, but without intervening. So even if there are 16 cells are produced, the size will be same to the zygote. That is the meaning of this thing. So due to the process of cleavage, many cells are produced, but without intervening the growth. So growth is not taking place, size is not increasing of the cell. Now, what is the goal? What is the purpose of cleavage? So cleavage is the first phase of the development, and due to the cleavage, large number of small cells are produced. Okay, and these cells are having high surface to volume ratio. So the cells are small in size, and this small size is helpful. Since the cell's size is small, they can easily uptake the nutrients. They can easily get the nutrients can easily pass through these smaller cells. Then oxygen is entering in the new cell, and then the waste material can be easily removed from this cells. Okay, now the third point. Now, due to the process of cleavage, many cells are produced, and these cells, they are acting like the building block of the embryo. So embryo, we can say embryo is made up of large number of cells. Once again, I have to pause this video because this noise is coming, so I'm pausing, sorry. So we were discussing this third point, that due to cleavage many cells are produced, and these cells, they construct the embryo. Now moving forward, this is the fourth point. Now imagine we want to construct a building, and the constructing a building will be very difficult for us if we try to build this building with a huge block of granite. So we cannot construct a building with a single block of granite. So similarly, embryo, for the development of embryo, this thing is important. Okay, now with the help of bricks, hundreds of bricks we can use, and we can easily construct the building. Similar way, with the help of large number of cells, we can easily construct the embryo, or we can say with the help of many cells the embryo is formed. Okay, and this purpose is fulfilled by the process of cleavage. So now all the steps of the cleavage. Okay, this is the figure, and one by one I am explaining all the points. So this is the first point. Okay, now 24 hours after the fertilization, the cleavage begins and it is completed about 6 hours later. So once again, the zygote begin to divide 24 hours after fertilization, and this first cleavage is completed about 6 hours later. So we can say 24 plus 6, nearly about 30 hours later, about 30 hours after the fertilization, the first cleavage division of the zygote has completed, and the first cleavage division produces two identical cells, and these smaller size cells are known as blastomeres. So in short, we can say after the 30 hours of the fertilization process, 30 hours after the fertilization, two small size cells are produced by the first cleavage division, that is mitotic one, and these two small size cells are produced, and these cells are known as blastomere. Now this mitotic division then produce 4 cell and then 8 cell, and this mitotic cell division goes on. So as you can see, from 2 cell then there is a 4 cell, then 8 cell, then 16. So at the end of second day, second cleavage division, which is a mitotic one, is completed and 4 cells are produced. So after the second day, 4 cells are produced. So this is the point. Okay, production of 4 cell. Now after the third day of fertilization, that is after the 72 hours, a solid ball of blastomere is formed, and this solid ball of blastomere is known as morula. So once again, after after the 30 hours, first cell division is completed and two cells are produced. Okay, then from this two cell there is a 4 cell, and this 4 cell, then there is a 8 cell production. Okay, so this is this 4 and 8, they are produced after 2 day of fertilization, and in third day, that is 72 hour, on third day the cell number is 16 or more. Okay, so the point is, after the first day 2 cell is produced, and between second and third day the number of cell is 4 and 8, and by the end of third day the cell number from 8 cell mitotic division, due to the mitotic division 16 cell are produced, and they are arranged, these all the cells, they are arranged like a solid ball of the blastomere, and this solid ball of blastomere is known as morula. So in other word, we can say the morula is a loose collection of cell, and it is produced. The blastomeres, they are arranged in a berry shape. The blastomeres are arranged like a berry shaped cluster, and in this cluster there are 16 or more blastomere cells, and all the cells together they are known as morula. So in the morula there are 16 or more cells are present. And what is the meaning of morula? So morula literally meaning little mulberry. Morula, morula. So the meaning of morula is little mulberry. Okay. So now we are moving forward. So once again, this is the zygote. Okay. Then from zygote, two cells are produced. Then there will be four cell. Okay. Zygote in this figure. From zygote, two cells are produced, then four cells, then 8, and then 16. So this 16 cell stage is known as morula. Okay, and this morula then it is converted into blastocyst. Okay, so now as you can see in the figure over here, in this is the morula, and it is converted into blastocyst stage, and we will discuss this blastocyst in the next lecture. And during this first phase of development, as you can see, all these new cells, okay, 2, 4, 6, they are traveling, they are transported towards the uterine cavity, and this one is the early embryo stage, and this is known as blastocyst, and this blastocyst is entering into, this one is the uterine cavity, and this blastocyst is going to implant on the endometrium. So during this entire journey, all these cells, whether they are 2 cell, 4 cell, or 8 cell, they are enveloped by, they are covered by zona pellucida, and the embryo finally reaches to uterine cavity, and for some time, for a day or two, it will swim. The early embryo swims, that is the blastocyst, it swims in the uterine cavity. So all these points you need to remember regarding this process of cleavage. I hope you have enjoyed this presentation lecture. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.